Okay, so can you hear me? Can you see this? Is it all fine?
Yes, everything's perfect.
Okay, so I'll try and go quickly through just a few more details about Corona Detective. I think some of you have probably seen something similar to this presentation before, but it could never have happened without the Open Public Community Lab where I work at Aquarium. Also, Fran Caro, who's now in Paris, but was in Madrid when this all started more than a year ago, and Guy Edelberg, who was at CRE, but is now in Haifa in Israel. Um, and we all came together to put together this diagnostic test based on isothermal amplification, as Keith has, and many of you have already worked with. I've been trying to make a 1-minute presentation about Corona Detective, and I think it's good that I'm trying to aim for 3 minutes now, but just we can talk more later in the breakout and so on, and you're very welcome to contact me at rachel@aquarium.ch. So we all came together during this pandemic in the quest for open science means to try and put an end to this pandemic. Of course, the Open Public Lab where I am, it's really has no real funding in particular. So without the funding of just one giant lab, we would never have been able to afford the molecular reagents, which we got everything commercially. All of our Corona Detective is currently based on the latest hot start BST2 from NEB and the RTX. That's also, actually they call it warm start enzymes. And we started very simply trying to make a lamp in dark COVID times. This is from Guy's thesis because LAMP is the simplification for this isothermal amplification that we use for this molecular amplification method. And similar to what Keith was talking about, we can get very sensitive and reliable detection of the virus down to 1 copy per microliter of reaction. And we're all about do it together research and democratizing research. And the big challenge for us right now is to see if we can get local production to happen. And so that's why it's so great to be part of this symposium. Thank you for asking us. So making this Corona Detective was, of course, inspired by Guy's GMO Detective, which he did his thesis work on in Ariel Lingner's lab at the CRE. And just as a quick overview, he did lots of workshops also with young students, old students, and everyone in between, to take food and search to see if there were unlabeled transgenic organisms like the commonly corn and soybean, that probably 90% of the soybeans grown in the world are transgenic. So with a very simple DNA extraction and this molecular amplification method, we could easily show people whether the control plant gene or the transgenic organism gene was in the unknown samples that people brought to these workshops. And using a very simple detector, you're able to very clearly detect if something's negative, like this first tube, or positive, like these next 3 tubes here. And the real key thing about GMO Detective and Corona Detective is that we put all these reactants together in a dried pellet format, so it can be shipped anywhere without reliance on the cold chain, similar to Sanchita's beautiful cellular reagents. So as Keith already mentioned, these methods are very sensitive. We get down to 1 copy per reaction. And the primer set we're using was already approved in 2 different EUAs that the FDA set up, the color one and the CRISPR one called Mammoth. So that's why we call it the NM primer set, because it's for the N gene of the virus for the Mammoth protocol. And here you can see we tried some early on environmental monitoring, swabbing doors, tabletops, and things like that. We never actually got any positive environmental samples. So we really think this is mainly useful for surveillance screening. And this 1 copy per microliter reaction is about 1,000-fold more sensitive than your average lateral flow devices where you're looking for the viral antigens. And because of this method of fluorescence detection that we used, a sort of quenched fluorescence, I thought I would skip all the details about that, but we have slides about it too that I can share. Basically, we can multiplex, so we can have an internal control rather than doing the side-by-side reactions like Guy did for the GMO Detective. We put it all in 1 tube, and so you can see the more green signal for the viral target and a more orangey signal with the hex tag for the cellular RNA internal control. And then both RNAs still show up on this very simple detector. And the future iterations of this multiplex could include looking for the flu virus, looking for other bacteria, other cellular RNAs, or maybe even the next pandemic cause, which of course should be expected. But the real thing we've been focusing on lately, and there's been a lot of back and forth with big companies, is how to do the at-scale production where all the regulatory agencies will be assured that each batch should be the same. So it includes for the lyophilization, the primers, including the tagged primers and a quencher. Nucleotides, of course, those have to also be RNase free, as the virus is an RNA genome virus. So you need to do the reverse transcription as well as the polymerization. And what we really hope is that these open possibilities for enzymes will mean that these reactions can be made much more inexpensively than when we're using these commercially available NED enzymes. And again, this fact that we can ship anywhere without the cold chain dependence still doesn't guarantee everything. For shipping, we still have tubes that were sent about 3 or 4 weeks ago waiting at the customs office in Sri Lanka right now. And so currently people in Ghana, some of you here in Chile, some of you here in Sri Lanka, people have tubes and are hopefully going to get maybe even regulatory approval in Ghana with the tubes that we sent them. And we're talking with many different regulators, and we're hoping that Reclone and local manufacturing might help a lot. And I should thank you for your attention, I guess. I don't know how I should have timed it better.
That's okay, it's perfect. So thank you a lot, Rachel.